Okay, people, in this yeah, okay, should I start that again? All right, in this week's video, we're going to be talking about polymerase chain reaction. And we're just going to get started this week on PCR. We're going to be doing more advanced stuff with PCR as we go along. But first, I wanted to start off with some work from last week. And so this is an example gel that you should have seen something kind of similar when you run yours. This is relatively well labeled. You can see here's our ladder. This is the size standard, and people have labeled out each of the individual sizes in the of DNA bands that you see in the standard. They've labeled here, this one's cut with HIN-D3, this one's cut with DD1. The thing that isn't so great is, well, it kind of looks like they didn't let the gel solidify long enough, and so the wells are a little wonky, and that's why you see some weirdness in the shape of the bands. They also didn't label which one was PGFPN1 and which one was PM Cherry N1. So that's one little issue. But aside from that, it's a nice clean gel and almost perfectly labeled. So what I want you to do for part two of this assignment is we see this what the plasmid looks like. Oh, the other thing that was wrong was they didn't run uncut DNA. We also want to make sure we're running some uncut DNA in this gel to see how that compares. So we want to know when we cut this DNA with HIND3 and DDE1, what should it look like? And so, if you see a pattern like this, does that mean that what you see is correct? Well, so what I've done is up on Moodle, I've given you the actual sequences and the DNA sequence for the PGFPN1 and the PM Cherry N1 plasmids. And I want you to individually paste them into this website. And so what this does is it runs your DNA sequence through all the known restriction enzymes, and it'll cut them up and show you how your DNA theoretically should be cut. And so when you go to run this, right under the paste box where you paste your DNA sequence in, click the circular sequence analysis, change that from linear, excuse me. And then towards the bottom, change the thing where it says only enzymes with recognition sites equal to or greater than X bases long. Change that down to 5 because remember from last week that DDE1 only recognizes 5 nucleotides, HIND3 recognizes 6, but if we put 5, then we'll get DDE1 and HIND3 in there. And so this is going to give you, it'll tell you where these DNA sequences are going to be cut by those enzymes. And so look through the list. It's going to give you a lot of different enzymes. But look through the list, see where your DNA will get cut, see what the sizes are supposed to be, and then compare what you see in this gel to what those sizes should tell you. And then in your post on Moodle, reply to your post and say, okay, this is what it should have been. This is what we see. Okay. All right. So remember this. Again, this week we're going to be focusing on the DNA level of life. Okay. So we haven't quite gotten the transcription and translation yet. So we're still focusing on DNA. So we have these DNAs, we run them on gel, we think they're kind of right, but what we're going to do this week is another test to make sure that these are actually the pieces of DNA that we think they are. And we're going to use a technique called PCR, and a couple of things we need to think about with PCR. Again, our backbone and most of the molecules held together with covalent bonds. The bases are held together with hydrogen bonds. And again, hydrogen bonds are relatively weak. They're more of an attractive force than an actual bond, but we still call them bonds. They're bonds. But basically, if we heat up DNA enough, the energy from the heat is enough to counteract the energy from these hydrogen bonds. And so what we can do is when we heat DNA up, we can separate it out into its individual strands. And that's really critical for polymerase chain reaction. The other thing that's really critical for polymerase chain reaction is the orientation of our strands. Again, we have a 3' end, right? and opposite, right? We go 5' to 3' on one strand, 3' to 5' on the other strand. They're anti-parallel, so the 5' and 3' strands are opposite from one another. And polymerase chain reaction is essentially DNA replication. And across all biology, whenever we're replicating DNA, we're going to be using an enzyme called DNA polymerase. And basically, any organism that's alive and some viruses that are technically not alive have DNA polymerases. Um, and one of the basically universal rules to life, and there are very few universal rules to life, but this is one of them. In all life, whenever DNA polymerase is copying DNA, it can only add a new nucleotide. So an individual base is called a nucleotide. And when it has 3 phosphates on it, it's called a nucleotide or nucleoside triphosphate. There should be an E on there. Um, Phosphates generally have energy associated with them. So you can think of the triphosphate form as the activated one that's ready to get put into a DNA strand. And so it uses free nucleotide triphosphates, or we call them DNTPs for deoxynucleotide triphosphates, DNTP. It uses DNTPs and the enzyme DNA polymerase to incorporate individual bases one at a time to complementary. So here we have an A, right? We're going to bring in a T, and the DNA polymerase is going to use that T and the phosphates, and it's going to create a covalent bond between this phosphate and the free 3' OH on this base. So in this case, we have a C there. It's going to add a T onto that C, but this T is complementary to that A. The next one, right? This one's a C, so the next base that would get put on is a G. After that, there's a G, so the next base that would get put on is a C, and so on. Okay, and this directionality, right? By adding to a free 3' OH, the overall directionality is that we copy DNA in the 5' to 3' direction. Okay, so there, for every DNA polymerase that we know of, basically it can't just hop on. Like if we ignore all of this down here, the DNA polymerase could not just hop on here and start copying this DNA. It needs a starting point. Okay, so it needs a free 3' OH to go and start adding more bases on. Okay, so that's one universal rule to life. So learn that one. We can only add new bases onto free 3' OH groups. Okay, and it's always going to be overall in the 5' to 3' orientation. And as I just mentioned, right, we can only add to something, to a starting point, and we call these primers. In life, in every organism that's alive, primers are RNA. RNA polymerase can basically hop onto DNA anywhere and start making an RNA copy of DNA. But life uses RNA polymerase to make little primers, and then the DNA polymerase can hop on to the free 3' OH group onto here and start copying the DNA strand in this orientation that we see here. Okay. You'll notice that this is an example of an origin of replication. So if we were to look at that plasmid DNA, right? Remember I mentioned that there's always going to be an origin of replication in plasmids. This is what it would look like when it gets copied. There's always going to be a leading strand that's very easy to copy. And actually for every origin of replication, for one orientation of our DNA strand, it's going to be a leading strand in this direction. And in this orientation of our DNA strand, it's going to be a leading strand in this direction. These are easy to copy. The lagging strand, since our replication has to happen in that 5' to 3' orientation, we get a primer, we get our DNA copied here, and then this thing down here where the DNA gets separated is called a replication fork, where we have to open it up and get new little pieces of DNA created with new primers. And so as this goes along, this little lagging strand process happens over and over and over again. And that's because, we can only add to free 3' OH groups. So this one's represented a leading strand here. This will be a lagging strand, and so this process would have to happen a bunch of times over and over. Okay, so one thing I haven't mentioned yet is that in life, we have an enzyme called helicase. Okay, so our DNA strand is normally double stranded. The helicase binds in an origin of replication and starts pulling the bases apart, right? So those hydrogen bonds that are holding our two DNA strands together, helicase pulls them apart and starts separating our DNA strands. When we do this in the lab, it's a little bit harder. We can't use helicase. And so this is what it really looks like during a DNA replication in an organism. And you don't have to know all these details, but what I'd like to know is it's complicated.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA...
Okay, so this right here is the helicase. And you can see here's one of our single strand piece, like, so here's our double helix coming into the helicase. Here's one of the individual backbone strands. Here's the other one. And these are the DNA polymerases, the purple things. And so you can see this is our leading strand. This one's getting copied really easily. The lagging strand has to occur in those little chunks. We call those Okazaki fragments.
To be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
All right, and so even though this looks pretty complicated, this is actually overly simplified, but it gives you an idea that, when you're copying DNA in organisms, you need a couple of DNA polymerases, you need a helicase and some other stuff. We don't want to watch that again. So if we want to copy this in a test tube, what do we need? Obviously, if we want to copy DNA, we need DNA polymerase. That's the enzyme that copies DNA, so that's critical. A primase might be nice, right? Something to provide a starting point for DNA polymerase. But this would also sort of lead to some non-specificity, because basically DNA primases can hop on anywhere and make little primers. And so what we do is we add our own primers, and our primers are made out of DNA, not RNA like they are in life. So we use little fragments of DNA that we call primers, and we can design primers ourselves. And based off of how we design our primers, we can amplify certain parts of DNA, like we're going to do this week. DNA helicase would be nice, right, to unwind the double helix, but it also adds some complexity. And the more proteins we add to a mix, the harder things get, right? So what we can do is use heat. Okay, so heat will melt those hydrogen bonds and allow the individual strands of DNA to be separated. But one of the issues is that heat can kill enzymes, right? If we took it, so to heat the DNA and melt it, we need to get it up to about 94 degrees Celsius. If you were to take human DNA polymerase and heat it to 94 degrees Celsius, it would get totally destroyed. Humans, we like our stuff at 37 degrees Celsius. And so obviously 94 is a lot higher than 37. And so that would destroy things. So we need a DNA polymerase that can withstand that kind of temperature. And so some genius along the way, I should probably know this person's name because it is an amazing discovery. They decided to try to purify the DNA polymerase from a bacterium called Thermus aquaticus. And this bacterium lives in thermal vents. Okay, so it's like totally cool with really high temperatures. And basically, if you take Thermus aquaticus DNA polymerase, we call this TAC, and heat it to 94, it doesn't care. You can heat it to 94 and cool it down, and it'll still work. And so basically, what we can do is the TAC enzyme is works really well at 72 degrees. And so if we have that enzyme at 72 degrees, it'll copy DNA at that temperature. If we heat it up to 94, we can melt our DNA strands and then let primers bind. And we do this cycling process. And what that allows us to do is copy DNA. Okay, so here's our 98 degrees. Here's our individual DNA strand up here, right? Our double helix. We're going to heat this up. This says 98 degrees, 94 degrees is totally fine. When we do that heating, it causes our double-stranded DNA to separate out into its individual strands. We cool it, and we have our primers in this mixture, and that allows our primers to bind. Okay, so somewhere between 48 and 72, generally we have our primers try to anneal at about 60 degrees Celsius. So our primers anneal to the individual strands of DNA. Then we heat that back up to 72 degrees Celsius. 72 is where our TAC is going to be active. The TAC is going to hop onto that free 3' OH, right? We have a free 3' end there on our primer. The TAC is going to bind and start copying our DNA in this direction. It's going to also, we're going to have a different primer in a different location. The TAC is going to hop onto that primer, start copying in this direction. So essentially what winds up happening is if we start with essentially one piece of DNA, by doing this 30 times, by doing this cycling process, right? Because if we take one DNA and copy it, we get 2 pieces of DNA, take those, copy it, we get 4, and so on and so on, right? We get an exponential amplification of our DNA. So by 30 cycles, we get 2 billion copies of our original piece of DNA, right? And it's only the pieces between our primers that get amplified by that number, okay? So this is a really great PCR video. I'm going to post this link on Moodle. I want you to watch this video. I'm not going to make you watch it right now with me, but this explains PCR really well. And so it is a requirement that you go and watch this video. So what can we amplify with DNA? Or with PCR. So we could amplify genomic DNA to find things like promoters and alu elements and different polymorphisms, so DNA base pair changes. We can use it to identify species with this kind of amplifying from genomic DNA. Ultimately, we're going to be doing some gene expression PCR, so we can check to see if genes are expressed, right? So that would actually be at the RNA level, essentially. We can clone things, which we'll be doing in a couple of weeks here. Then we can check for specific pieces of DNA out of big collections of DNA called libraries. And we can do DNA sequence to actually figure out what our DNA base pair arrangement is. So the PCR has become a really powerful technique. The tests that we're all getting for COVID are based off of a PCR technique. So, it just shows you some of the flexibility that this technique can provide. So what are we specifically going to amplify this week? So remember, this is our PGFP-N1 primer or plasmid. The PM cherry N1 plasmid is essentially the same thing. Instead of green fluorescent protein in here, it has cherry, which is a red fluorescent protein. So what we're going to do is we're going to use primers for this part. I mentioned this part before. This is the promoter that's active in mammalian cells. So we're going to have a primer that is the forward primer that corresponds to the promoter. Then we're going to use a reverse primer that corresponds to this thing called the SV40 poly-A tail. It's not important what that is, but basically we're going to be amplifying between here and here and amplifying. It's going to be the GFP and the multiple cloning site essentially that we amplify here. And so this one it binds at about 591, right? If base one is up here and we go around clockwise, the CMV forward primer binds at 591. The SV40 reverse primer binds at 1412. If we subtract this from this, we get 821 base pairs. So when you do a PCR using this primer pair, we should be seeing a PCR product, right? We call the piece of DNA that we've copied a lot of times, we call that the PCR product. Okay, we should see a PCR product of 821 bases. So what do we need to do PCR? Okay, so most kinds of reactions, right? We did a restriction digest. We had water in there. Water is going to be our main component of a lot of things. And, reactions need a place to happen. Okay, so then we'll have a buffer. So the buffer, just like with our restriction enzyme, the buffer provides pH. It provides some ions and other stuff that enzymes prefer their activities to be ideal in. We need our primers in order to get a real PCR reaction. You need both a forward primer and a reverse primer. And then we need our free deoxynucleotide triphosphates, or our DNTPs that I mentioned before. Again, these are the free individual bases that get incorporated into the new DNA strands. We need a template DNA, so a starting point. And of course, we need our TAC. We need our enzyme that copies our DNA. And so these are typically, this is just an example of a typical PCR reaction to show you what kind of things would go in there in a proportional amount. Ignore this sentence. I forgot to delete that. And so what happens when we assemble our reactions using all the stuff that I showed on the last slide, and then we stick it into a machine called a thermocycler or PCR machine. And what that does is it changes the temperature for us. Okay, so the initial 94 degrees does a melt, right? So that's going to melt our strands. We do an initial one that's a little bit longer to make sure all of our stuff's single-stranded. And then we enter the cycling phase. Okay, so we have our initial 94 for the melt. We go down to our annealing temperature. I'm showing this as 54, but I think yours are going to be 60 degrees. So that cooling allows the primers to bind. So if this is our whole big DNA strand, and our primer would bind to that, right? That annealing step is when the primer binds onto your single-stranded DNA. Then we have our extension time, right? So if our primer is now bound, we add our Taq polymerase, and then the extension is when the Taq polymerase is actively copying DNA. We go through these a bunch of times, 30 to 35 times, right? So we go back up to here, we heat it to 94, that causes another melting, go back to annealing, and so on, right? And this is the chain reaction part, right? Once we're done this 35 total times, we do a final fill in for 3 minutes, and then we hold it at 4 degrees. 4 degrees is refrigerator temperature, and so that kind of keeps everything stable. When we do PCR reactions, the two things that change most often are this annealing temperature, and that temperature is dependent on your primers. So depending on your primer composition, your annealing temperature is going to be slightly different. So the ones we're going to be doing this week, I think are I forget if they're 55 or 60 degrees. And then the extension time is based off of the length of the DNA to be amplified. Okay, so our general rule of thumb is 1,000 base pairs per minute. Okay, and so for this week, we're doing, I said 815 bases or something like that. So we'd need about, 45 to 50 seconds to amplify something that long. Okay, so you can make this as long as you need. You can change this temperature as much as you want, but most of this other stuff is generally kind of fixed. We're going to specifically do primers. We're going to design primers in a couple of weeks, but I wanted to give you a couple of primer design considerations just to start thinking about. Okay, so you're always going to need a forward and a reverse primer. And they need to be in separate places, right? You don't want your forward and your reverse primer to be right on top of each other, because then you don't actually amplify anything. You want them to be separate in the DNA, so that you wind up amplifying the region between them. Okay. Um, when we design our forward primer, it's the exact same as whatever DNA sequence we're looking at. Okay, so a lot of times we're looking at messenger RNA sequences. And so our forward primer is exactly the same thing as whatever we're looking at. When we do our reverse primer, it's always going to be the reverse complement of whatever we're looking at. And so if we see it, remember when I was showing you the restriction sites, right? We had the forward sequence, and then below that, we had the reverse complement. That bottom sequence is our reverse complement. So whatever sequence we're working with here, we take the reverse complement of that for our reverse primer sequence. And again, that allows the primers to point towards each other with their free 3' ends, and that allows the DNA to be copied. Okay. All right, so this week, what are you going to be doing? Exactly. So you're going to do 3 PCRs with your plasmid as the template in each. Okay, one reaction is going to have both primers, the CMV forward, the CMV 5' and the SV40 3', the SV40 reverse primer. One reaction is going to only have forward primers, and the other reaction is only going to have reverse primer. And what this is going to show you is that, well, if you assemble the reaction right, hopefully you only see something in this first reaction. And this just to prove to yourself that you need both a forward and reverse primer in order to see something in a PCR reaction. Then you're going to run a gel for your PCR products. Typically, our PCR reactions are about 25 microliters. You only need to use 5 microliters of that to run on a gel in order to see your PCR product. And then again, you're going to see this is separate. You want to go back and do that restriction digest map thing that I showed you in the very beginning of this presentation, to see if your cuts from last week make sense. All right, and so setting up your PCR reaction maybe takes a half hour to 45 minutes. Once you get really comfortable with this, it should only take you about 10 minutes to set up a PCR reaction. To actually run the PCR in the machine, it takes somewhere around 3 hours. And so if you have that long, you can wait around for it to be done. Again, you should sort of be used to running a gel now. And so, making up a gel, letting it solidify, and then running it, that probably takes, I don't know, 2 hours-ish. So theoretically, while this is running, if you want to do it all in one day, you can set up your reaction. And then while your reaction's going, you can make your gel and have it solidify, so that as soon as your reaction's done, you can mix up with some loading dye and run your gel. Okay. And that's it for this week.